We're going to recap what we started on Friday. We started the Calvin cycle, the light independent reaction, and then this will take you through Tuesday and Wednesday when I'm in Tampa at my training, and we won't have to play any catch up. You guys can keep doing this while I'm gone. If you weren't there, in the absent folder is this document for the light independent reaction. What do you want?
A notebook.
Okay. You're welcome. It was pointed out at the top box, just to correct that, to instead of it saying from the light independent reaction, that it says from the light dependent reaction, because we're taking up what we just did in the light dependent reaction and the thylakoid membrane. So that's what we're piggybacking off of. So what we filled in so far is from the light dependent reaction. We have the energy molecule ATP that was synthesized in the process, that's that really big word, photophosphorylation. We added another phosphate molecule onto the adenosine diphosphate molecule, making it adenosine triphosphate. And then we have the electron carrier NADPH. These two are going to go to the Calvin cycle, where it's the light independent reaction. The entire purpose of the light dependent reaction was to use the energy from sunlight to split our water molecule in photolysis so that we can have that hydrogen. Oxygen is a waste product. The purpose of splitting water is not to release oxygen. That is just the waste product. And we want the high energy electron from photoactivation of chlorophyll A, because that electron has a lot of energy with it. We're going to be seeing it used in some further steps in the light independent reaction, also known as the Calvin cycle. So to finish that entire light dependent reaction step up, the products of just that, there's two steps, right? There's the light dependent reaction in the thylakoid membrane, and then the light independent reaction that is occurring in the stroma. The whole purpose of light dependent reaction is to have NADPH, right? NADP is a high energy electron carrier, that literally is what it is, and ATP. Next slide, down here. Okay, so because I'm not there, I broke down these individual stages. There's 3 of them, and so just like zoomed in separate stages. And then on the following slides, as you see I have at the bottom, the next slide has the note page filled in for carbon fixation. For example, this stage is called carbon fixation. And we're only going to be looking in the stroma of the chloroplast. That's where our light independent reaction is happening. Okay, so we've 2 things so far come into play. What's going to start this cycle is this 5-carbon sugar. It's called RUBP. I believe it stands for ribulose biphosphate. And this is actually like in a molecular shape what it looks like over here on the right. Those dark gray spheres are the carbons. So it's a 5-carbon molecule. I think we're going to talk about things being 5. It's a sugar. Sugars are in a ring shape. 5 sides is like a pentagon. That's why I had you guys do a pentagon for it. You don't have to know what it looks like necessarily, but that's just one of the ones we're going to have to know. So this is going to be starting the Calvin cycle, RUBP. It's always starting it. We're going to have CO2 from the atmosphere come in this way now. And then we have Rubisco. Rubisco makes reactions go. They make reactions happen faster. They're enzymes, which means that they speed things up. We call those things catalysts if it's going to speed up a reaction. But when it's in our bodies, if it's biological, it's called an enzyme. So enzymes are biological catalysts. They are something that speeds up processes in living things. A pencil sharpener, an electric one, is a catalyst for sharpening a pencil. It speeds it up. There we go. So there's our Rubisco. It's an enzyme. It's going to catalyze reactions with CO2 and RUBP. So this stage, honestly, is going to have, I think, the most like amount of molecules or like players involved in this scenario. Okay, and so like visually, so I drew this little shape against the CO2 image and then the RUBP. If you remember from regular biology, enzymes fit this shape of the substrates, and the substrates are what's going into a chemical reaction. So I just drew it along the lines of this shape here so it could fit, and it's going to bring them together. And what do we get whenever we catalyze this reaction? So we're bringing together CO2 and RUBP. It's actually a very unstable compound, so it doesn't keep its molecule. Like it's unstable. It's going to break down pretty quick. We refer to this, we get to say this, not every class gets to say this, but for the sake of our class we get intermediate molecules. So molecules that are like in between of like our final molecule, which is glucose. So these intermediate molecules, there are further chemical reactions that are going to happen, like chemical changes that will happen, but they are not our business. And we're grateful for that. If we want to be nosy, it looks more like this. We don't need to know that. We don't need to know that. So I try to keep it as simple as we can. Woo! All right, so on the next page is where I have the note sheet with the box filled in. So this first step is carbon fixation, and we are catalyzing the reaction using Rubisco, an enzyme. And in my head, I think Rubisco makes things go, even though it's Rubisco. Rubisco catalyzes the reaction between RUBP, which is a 5-carbon sugar, and carbon dioxide. It's going to synthesize unstable intermediate molecules, these unstable sugars that are not yet our final product, but they're none of our business. And now our note page should look like this. If you were here on Friday, you may have had just these first 2 bullets written down. RUBP, a 5-carbon sugar, starts the reaction. Like it's always going to be the starting. Like we can't do anything with the carbon dioxide and Rubisco unless RUBP is there. Rubisco is an enzyme. It's going to combine CO2 with RUBP. It's a 5-carbon sugar. And then this was not written on Friday, but we have it now. It's going to create an unstable 6-carbon compound, or it just really creates, you don't need to know that it's a 6-carbon compound, but it just really creates unstable molecules, unstable sugars. And we see ATP and NADPH moving in from the light-dependent reaction. And the high-energy electron and its high-energy electron carrier, NADP and the H, are again moving into this process. Okay, and now this is referred to as the reduction stage. I should mention too, there's not any syllabus learning outcomes that ask you to know what the different stages are called. That first stage was called the carbon fixation stage. This one is called the reduction stage. That is just what they're referred to as, and it was a way for me to like mentally separate these different things going on. So you're not tested on those, but essentially within the name is what's happening. So still in the stroma, now we're going to use ATP. ATP is the energy molecule for the cell, and NADPH. Now those are actually coming into play, both from the light-dependent reaction in the thylakoid membranes. They're used to further like reform and change those intermediate molecules that are unstable. We can't really use them as a product. And reduce, which is taking an electron away. Just to refer back to the light-dependent reaction, remember there was something becoming oxidized, and oxidized, we looked at that more in the term of photooxidation. Oxidizing, we're losing, it's the loss of an electron. I'm gonna cough. Okay. It was chlorophyll A, that was photooxidized because of photoactivation, and it lost that electron. Here is that electron. This electron is going to be used to reduce those intermediate molecules. So that's why it's called the reduction stage. If you're taking on an electron, you're taking on negativity, becoming less, if you will, becoming more negative, and that's why it's called the reduction stage. But again, those molecules are not our business. There's our NADPH. So we need the NADPH because it's carrying the high energy electrons. That is its job from photoactivation, and it's there to reduce the intermediate molecules, the intermediate compounds that are made. And the hydrogen ion from photolysis is going to be incorporated into our final product, glucose. Finally, glucose is made. And this is the epitome of the carbon fixation. Photosynthesis is using light energy to fix carbon compounds into an organic molecule. And glucose is organic because it was made by something that is alive, and it can be used by things that are alive. Glucose is the building block of all like biomass, all food energy. And then from glucose, you can have other sugar structures made. Glucose is also what's used in respiration, and like it's just its simplest form, just what it looks like down here. We'll break sugars down like it's, you know, a completed Lego piece down into the simplest piece of Lego, and glucose would be like your simplest piece of Lego. And we see NADP, it did its job. It's going to go back to the thylakoid membrane, right? So if at this point we could just forget, could I make it bigger? I don't know. We can just forget that the NADPH is there. It's not there. It's done. It's used that electron to reduce these intermediate molecules. I did not include what that looks like because we don't need to know those molecules. And then it's used the hydrogen to synthesize the glucose molecule. So now we have this empty NADP. There's no H, it's in glucose. It's not full of energy, it's not carrying an electron anymore. It's going back to our thylakoid membrane so it can carry an electron again from photoactivation, carry a hydrogen again from photolysis. All right, and so we have building on to our note page for the reduction. ATP and NADPH are used to reform, or you could say not necessarily just reform, but also reduce, more importantly, reduce, because this is the reduction phase, those intermediate molecules, and that means that they're taking that electron, they're gaining negativity. Glucose is finally synthesized. That is the whole reason for photosynthesis. And we have this empty, lonely, low energy NADP molecule going back to the thylakoid membrane. And now we're in the final stage. It's the regeneration stage. So we're going to be regenerating something. So once glucose is synthesized, we're not finished. If we stop there, then the stroma or the Calvin cycle would never be prepared to do this again. So we need to like reset the stage for this process to happen again. Reforming molecules is going to require energy. And so the energy molecule for the cell is ATP. And what we're going to need to reform is the 5-carbon RUBP molecule that we started this entire process with. We started this process with RUBP and CO2, and those two molecules were catalyzed together by the enzyme Rubisco. So final stage, we need to reform the RUBP because RUBP is going to be starting the Calvin cycle or the light independent phase, and it's going to be the last thing that happens. It's going to be like reformed again so that we can have the cycle continue. And ATP is adenosine triphosphate, so you have 3 phosphates. When a phosphate is used, that third phosphate, when it's used, that's where all that high energy is located. Then essentially you have adenosine diphosphate. You only have 2 phosphates on that molecule now. It's not usable for the cell, so we have it going back as this like drained battery, essentially needs to be recharged. And by recharging, it's going to go to the thylakoid membrane and get that third phosphate put back on. And that's in that big process we added to our repertoire last week of photophosphorylation. So using energy from the sun to cause another phosphate to be put on this molecule. In your exam questions, this actually is labeled as number 4, but you have 2 number 4s because I'm always good for at least 1 mistake. This is so far the visual that I've seen for exam purposes of the Calvin cycle. It looks nasty, but we can kind of dumb it down for ourselves. So similar to what we saw with the light dependent reaction question, this is what it looks like. And notice where it says 6 carbon compound, 2 3 carbon compounds, another 2 3 carbon compounds. Those are those intermediate compounds that we don't have to know, which is why they're given to us. So what we do have to know, we can see as an example right here. So this first one, similarly to the one with the light dependent reaction, it says name the stage and state where it would occur within the chloroplast. And because carbon is involved, and we don't see any light being involved, this is not the light dependent reaction, this is the light independent reaction. Where it occurs in the stroma. I wonder if that's going to come back. Okay. Oh, it just goes away. That's perfect. It occurs in the stroma. The enzyme at AICE, the only enzyme that we have to know, it is Rubisco, and that is, again, that's starting this initial carbon fixation process. And if we look here, we have this 5 carbon compound. They don't ask you what it is, but the 5 carbon compound right here, that is your Rubisco. No, I'm sorry, that's the RUBP molecule. I'm talking and not thinking. That's again what starts this process off, and it's also what we're going to be ending with. It has to be reformed. So the compound at B versus the compounds at C. If anything with this question, those are what's a difficulty. So compound B is only used once, and then compound C, we see it being used twice. So the compound that's only used once, we only mention once, is the NADPH. So it's using those high energy electrons to reduce those intermediate compounds, and the H on the NADPH is going to be incorporated into our final product. C gets used twice. So you have a molecule ATP, it's going to be ATP, that is used to essentially add energy so that we can be reducing and reforming and adding that hydrogen on, just having a chemical reaction occur. And then over here, C is used because we need to reform and regenerate, that's the regeneration stage, the RUBP molecule, so we can start this whole process off. Compound D is, notice it's leaving this entire cycle. It's the only one that's really pointing like completely out. It's not just curved to go back in somewhere, it's pointing completely out. That is the glucose molecule, the whole reason why we're doing this. Okay, so here we can see kind of all the factors involved for the regeneration phase. You can see I've filled this in for you. ATP is used to regenerate, reform REBP to start the cycle again. It starts and ends with that. And ADP, love the tired emoji, not much energy. It's going to go back to the thylakoid membrane for photophosphorylation. And pause where you need so that you can write this down. If any of the previous slides add more context to what you're writing in the boxes, I think you can use the back of this note page to add some of those for context for yourself. And certainly complete that exam question. Okay, so this is the entire light independent phase with all the steps included and all of like our little molecules involved in the picture. Okay, and it's all going to start again. So again, notice in the last slide, ADP and NADP. So adenosine diphosphate's missing its energy molecule, that last energy molecule of phosphate, making it a triphosphate. It's missing that, it was used. And NADP, no electron on there and no hydrogen on there from the light independent reaction are now going to enter the light dependent reaction again to be refueled as energy molecules for the Calvin cycle. We call the Calvin cycle the light independent reaction. This is what we've already completed, the light dependent, and we have some of the leftover molecules from the light independent. So it's all a cycle. We started with light energy and it caused chlorophyll to become photoactivated, which means it lost this electron. That electron is going to be carried on this NADP molecule. I'll call it H in a second. We have water entering the light-dependent reaction. The energy from the sun is going to cause photolysis, where we're splitting that molecule up. Oxygen is released as a waste product. And now that hydrogen ion is going to be incorporated in the NADPH. They will travel over to the stroma for the light independent reaction or the Calvin cycle. But without reiterating the entire light independent reaction, the only thing that I've added on is the leftovers from the light independent reaction. So right here, we have the NADP, no H, no electron that was used to synthesize glucose already, and it used the electron in the reduction phase. And we were reducing those intermediate molecules that we don't need to know any further about. And our ATP molecules were used to give energy to those different reactions in the Calvin cycle. So we see just a plain Jane ADP moving back into the light dependent phase. I think hearing this multiple times is helpful. So if you need to replay it, then replay it. But this will pretty much be the end of like the lecture of these different phases in photosynthesis. And if you have those notes done, listen to this a couple times. I think this is the most challenging part of the syllabus. But as we all know, repetition helps, and we've done it. Yay! Okay, I'll see you guys soon.